Chapter 11, Cancer. Here are the primary objectives for this chapter. You can go ahead and pause the video and read these to yourself if you would like. Okay. Section 11.1, Cancer cells develop in distinct ways. So as of this moment, cancer is still a pretty scary concept, but it will continue to get less and less frightening as scientists continue to make advances in cancer treatment. I'll even go on to say that we're pretty much winning the war on cancer at this point, and the reason I say this is because 5-year cancer survival rates have been rising since 1970 from about 50% to about 70% today, and the 5-year survival rate for specific types of cancer is even higher than 70%. This is just the average survival rate for all types of cancer. Our bodies actually produce cancerous cells each and every day, but most of these cells are killed by our immune system, and actually I'll even go on to say that virtually all of these cells are killed by our immune system. Cancer cell production within an individual is actually a pretty rare event, relatively speaking. Cancer is not one single disease, but is actually more than 100 specific diseases, and each of these forms of cancer requires a specific form of treatment. And as treatment regimens are refined, each individual's treatment regimen becomes a personal plan to fight their individual specific cancer. Cancer cells have certain characteristics which explain why they behave the way they do. Cancer cells have no control over their own growth. Cancer cells lack differentiation, which means they have absolutely no specific function in the body and absolutely no homeostatic function in the body. They have abnormal nuclei. Their nuclei are typically larger than those of normal cells, and they also have abnormal chromosomes. Cancer cells are also immortal, which doesn't mean that a cancer cell lives forever. It just means that it has an unlimited ability to divide. So we're actually talking more like the cell line is immortal. And these immortal cells have unlimited potential to replicate, and they can avoid apoptosis or cell death, because normally when a cell has a mutation which can damage its function, it will actually commit apoptosis and kill itself in a manner of speaking. How do cancer cells avoid apoptosis or cell death? Well, the answer to this question is related to the fact that normal cells have both growth-stopping genes and growth-promoting genes. And the fact that cancer cells can avoid apoptosis is actually related to the fact that they have no control over their own growth like normal cells do. So in your normal somatic body cells, you have these genes called proto-oncogenes, which are growth promoter genes, and the suffix onco usually means or is related to cancer, so it's saying proto-cancer genes. And these proto-oncogenes are regulatory genes necessary for normal cell growth. And I'd like to point out that at this point, these genes actually aren't cancer genes, but they have the potential to become cancer genes if we experience some sort of genetic mutation within these genes, such as a deletion of a base pair or an unnecessary addition of a base pair. And then normal cells also have what are called tumor suppressor genes or growth inhibitor genes, which are the genes responsible for initiating apoptosis of a mutated cell that can't function properly. And one of the most important types of these tumor suppressor genes is a gene called TP53, and this gene encodes a protein called P53. The protein is responsible for halting cell division in abnormal cells unless or until damaged DNA is repaired. So pretty much the job of this protein is to be saying, hey, unless you fix this damaged DNA, we're going to have to kill ourselves. And if this DNA cannot be repaired, P53 initiates cell death or apoptosis. And it's estimated that more than 50% of all cancers involve some sort of faulty P53 gene. And if both the tumor suppressor genes and the proto-oncogenes in the same cell experience random mutations leading to an avoidance of apoptosis and the cell's inability to control its own growth, this could possibly result in cancer. So this picture just shows what happens when a normal cell experiences a random mutation compared to a cancer cell experiencing a mutation. So at the top we have the normal cell, and it has damaged DNA or mutated DNA that it cannot repair. And since this is a normal cell, it has an activated P53 gene such that it's producing these P53 proteins. And since it is not capable of repairing this damaged DNA, the P53 proteins cause the cell to commit apoptosis or cell suicide to ensure that we don't pass on this damaged DNA to the next generation of cells. And compare this to the cancer cell at the bottom, which has both mutated proto-oncogenes and a damaged P53 gene, which means if it experiences any more damage to any other types of genes that will make this cell more malignant, it will not be able to commit apoptosis because this P53 gene is damaged and it's no longer making P53 proteins. So this cancer cell just goes on to continue to divide despite having this damaged DNA, and it'll pass on its mutations to the next generation of cancer cells. There's another way cancer cells avoid apoptosis, and it's related to obtaining immortality. So within our cells we have these links of DNA called telomeres, which are actually stretches of repeating DNA bases located at the tips of chromosomes. And these telomeres are maintained by an enzyme called telomerase, which the body stops producing soon after birth. And each time a cell replicates, a little bit of this telomere is snipped off because we don't have telomerase at this point to repair it. And it's estimated that 50 to 60 divisions of a cell, and by that time there will be no more telomeres in that cell's DNA, and at this point a cell stops replicating and dies. However, if telomerase is somehow present in a cell for whatever reason, the telomere is repaired after every division, and the cell has essentially obtained immortality, and its line will live forever. And this is what occurs in some cancer cells. Now, if the TP53 gene is faulty, or the cell continues to produce telomerase, like I said, the cell can also replicate rapidly and uncontrollably. And yes, this is how the cell achieves immortality. It does not receive, nor does it respond to apoptotic signals anymore, and it is out of control at this point. So now that we have this line of cells which has obtained immortality and has the potential to divide exponentially, the cancer cells are now going to multiply and divide to form tumors. And a tumor is a growth formed from a grouping of cancerous or precancerous cells. And these tumors form when the cells lose their natural inhibitions and begin to grow and then pile up on each other. And this is typically not the case in normal cells because they can send chemical messenger signals to each other to tell each other, hey, we've already grown as much as we need to grow in this area, so let's quit growing at this point. But anyway, tumors can be benign, or rather, they're called benign or malignant. And these malignant tumors are harmful and invasive, and they're capable of spreading into adjacent or other tissues. And then we have the benign tumors, which are the precancerous and harmless tumors that do not spread. And these include moles, polyps in the colon, and warts on the skin also. And over here in this picture at your right, the picture's showing how cancer cells begin to pile up on each other. So at the start of it, we have this one genetically altered epithelial cell. So this is going to be a cancer of the skin. This altered cell then begins to multiply, and once it's multiplied to a certain extent is where we have this benign tumor in this case that could potentially become malignant if it experiences any more DNA mutation or random mutation of any kind. So just to reiterate what I've already said in the past slides, cancerous tumors form when proto-oncogenes and tumor suppressor genes mutate. So when proto-oncogenes mutate in a certain way, they form oncogenes or cancer genes, and these cause cancer, of course. There are many proto-oncogenes in each and every cell in the body, so it is possible to form many oncogenes in one cell. Every oncogene has the potential to disrupt cellular function, so the more oncogenes that are activated and mutated, the worse off the cell will be and the greater its potential of becoming a malignant cancer cell. Tumor suppressor genes can also mutate and lose their function, of course. Altered tumor suppressor genes will no longer regulate the cell cycle and cannot promote apoptosis. And all of these mutations, either proto-oncogene mutations or tumor suppressor gene mutations, they're all called loss of function mutations because the cell has lost the capability to function properly. And if a cell winds up with extra copies of oncogenes or not enough tumor suppressor genes, or if both of these occur, it may become a cancer cell at this point. So our body actually produces cancer cells every day, but this isn't usually a problem because the immune system typically does a pretty good job of destroying cancer cells. And the reason we produce cancer cells every day is that DNA is replicated continuously, which results in a constant occurrence of cancerous mutations. Thus, we produce cancer cells every day. But thankfully, we have an immune system that does battle with cancer cells every day. And the immune system accomplishes this because many cancer cells have antigens on their surfaces that are not found on normal cells. So then the immune system can essentially use its ability to recognize self from non-self, and they then recognize these cancer cells and kill them. And the cells responsible for taking care of cancer cells are T cells and natural killer cells. However, some cancerous cells have developed or do develop ways of resisting the immune system. Some cancer cells may develop some mechanism of avoidance just so they can avoid the immune system, or a group of cancer cells have developed the ability that they grow so fast that the immune system cannot possibly destroy all of them. Also, if the immune system is compromised or otherwise weakened, cancer can then gain the upper hand in these situations sometimes, or many times, depending on how you look at it. Cancer progresses in stages, but actually starts with only one cell. Every tumor starts as essentially one cell gone wild. And these cancer cells must compete with other normal cells for nutrients and space. So, in order for a cancer cell to survive, it must have some sort of advantage over its neighbors, such as apoptotic resistance. And an advantage like this would ensure a cancer cell's survival, allowing it to pass on its traits to its descendants, though in this case, it would still have to avoid the immune system also. The cancer cell descendants then tend to accumulate even more mutations, making their progeny or descendants even more abnormal. These mutations could then allow the cells to continually change with each generation, and it will make it much harder for the immune system to track down and destroy them all if this occurs. Once cancer cells have successfully overcome the immune system, they can continue to grow and multiply unchecked. They can also then successfully outcompete normal cells for space and nourishment within the body. And this signifies stage 1 of carcinogenesis, which is cancer development. And this isn't the same type of stage that physicians assign to cancer. This is more like the stage of cancer development. So the stages that the physicians are assigning don't have that much relevance on the actual development of cancer. And we'll discuss why in a second. So once the tumor contains about 1 million cells, or is 1 to 2 millimeters in diameter, the interior cells of the tumor can no longer receive enough nutrients, and these cells begin to die, depositing their waste products within the cell cluster of the tumor. And this is called a carcinoma in situ, or cancer in place. And this means that in order for the cancer cells within the tumor to survive, they're going to need more blood. So, in order to accomplish this, the cells release angiogenic compounds, which are compounds that will lure blood vessels into the tumor. And the growth of this new network of blood vessels is called angiogenesis. The tumor is now effectively immortal and has begun to crowd out the surrounding non-cancerous cells, and it is capable of killing its host if left untreated. Cancer cells have the capability of invading almost any tissue. So not only do cancer cells grow uncontrollably and adhere to surrounding cells, they also tend to invade normal tissue, and they can invade any kind of body tissue. Once a primary site, or where the site where the tumor has initially grown, has been established, the cancer cells can then break away from the original tumor mass and travel through the bloodstream or lymph, and this is called metastasis. The migration of living cancer cells from the original tumor is called metastasis, and this is the mechanism by which cancer cells travel throughout the body. And these traveling cells are eventually deposited at secondary site where another tumor can develop and grow. So here are the stages of carcinogenesis or cancer development, and these aren't the same as the stages that physicians assign to cancer. One difference is there's 5 stages here, and there's not always 5 with the other types of cancer stages. And also here we're more referring to how we get a cancer cell in the first place, and less about how we're going to have to treat that cancer, which is a pretty poor explanation, but we're just going to go with it. But anyway, here are the 5 stages of carcinogenesis. Hyperplasia, which is where we have abnormal growths which contain excess number of cells. Though cell growth is unregulated, the tissue remains relatively normal. So also here in these stages, we don't have cancer cells in every stage because in hyperplasia they're not even cancerous at this point. Then there's metaplasia, which is when when the normal occurring cell layer is displaced by cells of a different type that are not normally found within the particular tissue. And this usually occurs in epithelial transition zones. So essentially here we're just saying they have one type of cells from one zone within the epithelium moving to another zone. Then there's dysplasia, which is the transitional state between benign growth and malignancies. Here the cells display changes in nucleus size and shape. So they now have a larger nucleus with an abnormal shape. And they experience increased mitotic activity and changes in their cytoplasmic features. So here they're basically just growing at an increased rate is what mitotic activity is referring to. And at this stage we're just transitioning between benign and malignant. The fourth stage is called in situ cancer, and this is the stage where the cells display high levels of dysplasia, and in this stage the risk of cells becoming cancerous and invasive is very high. And then finally we have these full-blown invasive cancer cells, where the cells now have the ability to metastasize and spread to other areas of the body, usually via angiogenesis. So here it's just saying that these cancer cells have access to blood vessels, which allows them to move to other tissues. Here are the stages of carcinogenesis from your book, and this picture doesn't show the stage of metaplasia, so we're just going to go through it anyway and skip it. So here at this first stage, which isn't actually a carcinogenic stage, we have this one genetically altered cell within the epithelium. So then this altered cell maybe replicates a little bit unregulated to where we actually just have more of the cell than we need, and it's formed a growth here. And then skipping metaplasia, we go to dysplasia, where the cells have become abnormal and are transitioning to becoming cancer cells, and we've begun to form this tumor here. And then we have in situ cancer, where the cancerous cells in the center of the growth multiply rapidly, but they run out of nutrients to grow, so they eventually die, to where we have this tumor mass of living cells surrounding all this dead cell material. And then finally, at the last stage where we are experiencing metastasis and we have a malignant tumor, the cancer cells are breaking out of the tumor. They're breaking out of this basement membrane in the epithelium, and they're coming in contact with a blood vessel, and then they travel to another tissue of the body and set up another tumor site. And I just need to say about the last slide, I actually got that from a homework assignment I had in grad school a few years back, so that's why it doesn't necessarily align real well with what the book says, but I think that it's going to be better for you to learn from that because it goes into more detail than what I think the book should have. So I think the best way to tell the difference between these stages of carcinogenesis and the traditional stages associated with cancer is just think that these stages of carcinogenesis are the stages that must occur for a fully functioning group of cancer cells to develop, whereas the more traditional stages here we already have cancer cells and we're trying to treat them. So the carcinogenesis is all about development, and the other stages are more about cancer that we've already developed. But anyway, moving on. Section 11.2, cancer has many causes and effects. There are 4 major categories of factors which can cause cancer, and these are heredity, environmental factors, viruses, and diet. So I'm sure you already knew this, but certain genes are linked to certain cancers, and there are a number of genetic markers which can predispose an individual to one or more forms of cancer. So the earliest discovery of a genetic association with a particular cancer occurred in 1990 with the discovery of these breast cancer genes. The first of these was breast cancer gene 1, or BRCA1, and then a little bit later on another gene, breast cancer gene 2, or BRCA2, was discovered. And for an individual to have breast cancer developed from mutations in these genes, a mutation has to occur on either both copies of BRCA1 or both copies of BRCA2 to develop breast cancer. So you can't develop breast cancer in this case if you just have one mutation on BRCA1 and one mutation on BRCA2. And generally, if an individual even has a mutated copy of one of these genes, they'll only have one inherited copy. And another mutation must occur later in life in order for them to develop cancer. There's also another gene, well, actually there are many genes obviously, but anyway, this other gene called the RB gene, which is a tumor suppressor gene, has been linked to developing retinoblastoma, which is a cancer that occurs in the eye, on the retina. And both copies of this gene must contain a loss of function gene for this mutation to occur. So pretty much just the same thing with the breast cancer genes. Maybe an individual is born with a mutation on one of these genes, but they're going to have to require another mutation on the other copy of the gene to develop cancer. The risk of contracting thyroid cancer is generally enhanced if the individual has a mutation in one of the two copies of the RET gene. So the same thing here, you need two mutated copies of one gene to develop cancer, or to have the potential for developing cancer. So you may not know what this diagram is below, just because in this class they're not really letting us go too far into genetics, because there are other things we're more interested in. But anyway, this is a pedigree chart, and this chart in particular is showing us a family with a predisposition for breast cancer. And all the circles are females and all the squares are males. And all of the filled in circles are showing a female with cancer, and the filled in squares are showing a male that's a carrier for a cancer, or rather he carries this mutated gene but hasn't developed breast cancer because he's a male, though I'd like to point out that males can get breast cancer, though it's rare. But anyway, in generation 1 we have this woman who has children, and then maybe she gets cancer later on, or maybe she gets it before she has children, it doesn't matter. But anyway, she has children that either get this cancer or they carry this cancer. So she has a daughter that has this cancer, and then a son that is carrying the gene that predisposes the family for cancer. But anyway, the son has children with a female who isn't carrying this cancer gene, and this gene is passed on to his two daughters, shown in red, which have developed the cancer. And then in generation 4, this female has children with a male not carrying the gene, such that she also has a daughter with breast cancer. So here we're just showing how this type of breast cancer is, or runs in a family. So in addition to heredity, environmental carcinogens can cause cellular mutations which can lead to cancer. And a carcinogen is any agent in the environment that can cause cancer. And they can act in two ways, and some carcinogens can actually act in both. So we have an initiator, which is a carcinogen that causes cancerous changes in cellular functioning. And then we have a promoter, which is a carcinogen that increases the likelihood that an initiator will affect cell functioning. And these carcinogens are all around us in the air we breathe, the water we drink, and the products we use in and around our homes. Some of these may be easy to avoid, while others may not be. A variety of naturally causing chemicals are actually carcinogenic. These include tannins in tea, safrole found in cinnamon, and a flavor-enhancing chemical in pepper. And typically, we can probably deal with this sort of stuff just with our immune system alone. But if you're eating just like a ton of any of these, it might be a good idea to stop, I guess. It's estimated that more than 50% of all cancers are caused by environmental carcinogens. So now we're going to talk about some chemical carcinogens which do not occur naturally, and a lot of these are found in tobacco smoke, which contains N-nitrosonar nicotine, vinyl chloride, benzoapyrimidines, and other chemicals, all of which can cause cancer. It's estimated of the percentage of cancer deaths linked to cigarette smoke run from 30 to 80%, and these include deaths due to secondhand smoke, which is quite terrible because you can avoid smoking pretty easily. And it's even worse if you're getting a lung cancer and you don't even smoke. And only about 2% of cancer deaths are linked to exposure to industrial pollutants, for example, asbestos. Pregnant women and their fetuses are especially susceptible to some kinds of industrial pollution. One other thing about industrial pollutants, you're not really going to encounter them unless you work in a job with that carcinogen. So most of the time you wouldn't have to worry about them as compared to some other chemical carcinogens. Another type of carcinogenic agent is radiation, and there are two types of radiation. There's non-ionizing radiation, which has no potential of causing cancer really. And an example of this type of radiation is electricity, and they've proposed in the past that electricity has led to certain types of leukemia, but none of these studies have really been proven, nor were they all that scientific, honestly. But the type of radiation we need to worry about in regards to cancer is ionizing radiation. And this type of radiation includes sunlight, or more accurately, UV rays. And typically the sun rays we need to worry about are UVB rays, which have been found to cause over 80% of skin cancers. And fair-skinned people are typically more susceptible. And then there's radon, which is a colorless, odorless gas that is released by water, soil, and rocks in varying amounts and intensities in different geographic areas. Home buyers should have their new home inspected and have radon detectors installed. There's also nuclear radiation, which can cause cancer, but unless you've been in an area that's been hit with a nuclear bomb recently, you're really not going to be exposed to this. And there's also X-rays, which are a type of radiation that can lead to cancer, but there's a very slight risk unless you have just gotten just tons and tons of X-rays, I suppose. This is a diagram of the electromagnetic spectrum, and it's showing these various types of electromagnetic waves that may or may not cause cancer. And as you can see with these arrows, as frequency increases, wavelength decreases, and vice versa. But anyway, in general, waves with a lower frequency are less likely to cause cancer, and these include radio waves, television waves, microwaves, radar waves, infrared rays or heat rays, and all the light in the visible light spectrum. And then we go on to types of waves with a higher frequency, such as ultraviolet light, X-rays, and gamma rays. These could all potentially lead to cancer, though In the case of gamma rays, they'll probably just straight up kill you first, I would imagine. Another factor which can lead to cancer development are viruses. And in the past, this wasn't as talked about as it is now, but this has changed with the discovery that the human papillomavirus is linked to cervical cancer. So, in addition to the human papillomavirus being linked to cervical cancer, there are some other viruses linked to other cancers. The Epstein-Barr virus, or EPV, has been linked to Hodgkin's and non-Hodgkin's lymphoma. Hepatitis B and C have been implicated in liver cancer. HIV has been linked to non-Hodgkin's lymphoma and Kaposi's sarcoma. And then finally, the T-cell leukemia lymphoma virus has been linked to T-cell non-Hodgkin's lymphoma. And we actually do not know exactly how viruses initiate cancer, but we do know that they insert their foreign DNA into the host genome and interfere with the host cell functioning. And in general, whenever foreign DNA is inserted into the DNA of a host, there's the potential that the host may develop cancer. And anyway, this inserted DNA can promote the development of cancer, possibly by adversely affecting the immune system and altering the balance between cancer cells and immune cells. Certain diets may also contribute to cancer, and actually being obese in general can contribute to cancer, because obesity has been linked to a 50% increase in colon cancer in both men and women, and a more than 50% increase in breast cancer and uterine cancer. And it's kind of obvious why it could lead to an increase in the risk of breast cancer, because just the more tissue you have up there, the more likely you are to develop cancer. Traditional Western diets have been linked to cancer, and these are diets high in fats and red meat, but low in fruits and raw vegetables. And this type of diet can lead to reoccurring colon cancer. But the good news about colon cancer is it's pretty slow growing, and we have techniques to deal with it, I guess. Nitrates, which are used as preservatives in some foods, have been linked to cancer, and these can be converted into nitrosamines in the digestive process, and that's the actual chemical that can lead to cancer. And like I said, these are used in preservatives in luncheon and smoked meats and other foods, and I believe the news has probably recently covered this. And these nitrates can increase one's risk of colon and other cancer. And then finally, well, not finally, but we have heavy metals and other chemicals found in fish and mollusks. And in this case, we're talking about mercury for the most part. And the types of fish and mollusks which contain these chemicals are fed in waters contaminated by chemicals or heavy metals. So it might be a good idea to limit the consumption of fish caught in coastal waters. So not really a concern in Missouri since we're landlocked, unless you are eating coastal water fish. And you need to especially keep away from those fish which are fatty, like sea bass and bluefish. And that's because fat's going to store these chemicals within it. And also, excessive alcohol consumption has been implicated with cancer, especially liver cancer. And it's also been linked to cancers of the mouth, throat, esophagus, and breast. We can also eat certain foods to help guard against cancer, so maybe we kind of counterbalance the supposed bad foods against the good foods. And anyway, as some foods are thought to promote cancer, other foods are probably going to help guard against it. And such foods include green leafy vegetables, which contain a precursor to vitamin A called beta-carotene, which acts as an antioxidant. Antioxidant vitamins in general counteract the effects of the free radicals that are a normal byproduct of cellular metabolism. These free radicals are generally detoxified by the body's natural processes, but if that cleansing is inefficient, they can build up in the body and damage DNA. Another example of an antioxidant is vitamin C, which is found in citrus fruit. Cancer can strike almost any part of the body. So here we're going to begin talking about the different types of cancer. And here are just some more technical names for the different types. There are carcinomas, which are cancers of the epithelial tissues, and these would include skin, breast, liver, lung, prostate, and intestinal cancers. There are adenomas, which are cancers of the glandular tissues, and these would include cancers of the thyroid or adrenal gland. There's adenocarcinomas, which are cancers of the glandular epithelial tissues, such as the adenocarcinoma, which occurs in the uterine cervix. There are sarcomas, which are cancers of the connective and muscular tissues, and these include bone cancer or osteosarcoma, muscle, and other fibrous connective tissue cancers. There are blastomas, which are cancers of the embryonic tissues, such as retinoblastoma, which occurs in the retina of the eye. There are leukemias, which are cancers involving the blood. And then finally, there is lymphomas, which are cancers which involve the lymphatic system. So here's a diagram of some classes of cancer and where they can occur in the body. So up at the top left, we have an adenoma in the form of thyroid cancer. There's breast cancer, which is a carcinoma. There's cervical cancer, only found in the cervix, obviously, which is an adenocarcinoma. There's leukemia, which occurs in the blood. Skin cancer, which is another type of carcinoma. There's Hodgkin's, which is a type of lymphoma, or Hodgkin's disease more correctly. And then there's a sarcoma in whatever connective tissue it's going to indicate. So now we're going to move away from the classes of cancer and move on to the individual types of cancer, starting with lung cancer, which is a carcinoma. And the diagnosis of lung cancer is based on symptoms. There's actually no screening test for lung cancer. It is diagnosed based on symptoms which include persistent cough, frequent pneumonia or bronchitis, or changes in the voice such that it becomes deeper and raspier. Cigarette smoking is by far the leading factor in the development of lung cancer. Other risk factors include household, workplace, or environmental exposure to chemicals, asbestos, and radiation. And in the United States, the 1-year survival rate is under 50%, and the 5-year survival rate is only 15%. So lung cancer is pretty deadly. So in order to prevent this, early detection is advised because it improves these rates somewhat. And lung cancer actually kills more people than the 5 next most deadly cancers combined. And this kind of makes me mad because cancer due to smoking is so preventable. Though I'd like to point out that some people do get lung cancer due to heredity. Colorectal cancer is another type of carcinoma which occurs in either the colon or rectum or both. And this cancer starts as benign polyps, which are just tumors within the colon, benign tumors. And the screening test for this is the colonoscopy. An initial colonoscopy at age 50 is recommended for most people, and some people will need earlier screening if they have a greater risk. So here they just would have some sort of genetic predisposition. A colonoscopy is done under sedation and involves snaking a fiber optic tube and camera through the anus and the length of the colon to the junction of the small and large intestines. And this sounds really awful, but if it's going to keep you from getting colon cancer, I can understand why they would want you to do this. Rectal bleeding or bloody stool, even in a younger person, may necessitate a colonoscopy just so they can rule out cancer. And generally, a high-fat, low-fiber diet and sedentary lifestyle is linked to colon cancer. And like it said, colon cancer starts as a benign polyp or tumor that protrudes from the lining of the colon. One good thing about these polyps is that they grow slowly and usually never develop into malignancies. However, early detection is important. The 5-year survival rate for colon cancers is currently about 64%, but the 5-year survival rate after the cancer has spread is only 10%. So that's kind of another reason why you ought to get a colonoscopy at age 50 and beyond. So here's an image taken during a colonoscopy at your right, and you can see the polyp clearly labeled there. So it's just the cellular growth, and usually over the course of a colonoscopy is when they'll actually cut off this polyp so it doesn't grow into something malignant. Also, I forgot to tell you from the last slide, but if you're 50 years or older, it's recommended that you have a colonoscopy every 10 years, whereas if your family has a history of colon cancer, you may need a colonoscopy every year or every couple of years. There are several risk factors for developing breast cancer. Breast cancer is almost solely a woman's cancer, although about 1,700 cases of male breast cancer are diagnosed in the U.S. each year. Breast cancer is age-linked. It's estimated that 1 in 200 women will develop breast cancer before age 40. 1 in 26 women will develop breast cancer before age 60. So we're seeing a clear correlation with breast cancer and age here. Other risk factors include early menarche, which is when a woman begins to go into menopause, occurring earlier than normal in this case. Late menopause, obesity, especially after menopause, and use of hormonal contraceptives and hormone replacement therapy after menopause. So the way that breast cancer is diagnosed is through a mammogram, and we'll talk more about that in a second. In the U.S., the 5-year survival rate for women with tumors that have not yet spread is 98%, so that's really good. And the overall 10-year survival rate is around 80%. So we've pretty much got breast cancer on the run, it sounds like. For those with metastatic breast cancer, which has spread to the lymph nodes, however, the 5-year survival rate is much lower. So essentially, if you can get it found or diagnosed early, your chances of survival are pretty high. A mammogram is essentially just an x-ray of the breast tissue, and here's a mammogram machine. And honestly, this whole thing looks kind of painful, but it'll help to prevent cancer. And anyway, in a mammogram, the physician's looking for any lumps of tissue within the breast that might be cancerous. Another type of carcinoma is skin cancer, and there are 3 types of skin cancer. There is basal cell carcinoma, which affects the basal cells of the epithelium. There's squamous cell carcinoma, which affects the epithelial cells produced by the basal cells. So these are the cells above the basal cells. And both of these appear as small abnormal patches on the skin. They spread relatively slowly, but should be removed to ensure that they don't do us harm. And then the third type of skin cancer is melanoma, which occurs much less frequently than the other 2, but this cancer is capable of metastasizing very quickly and is thus much more dangerous. However, if melanoma is caught early, the 5-year survival rate for those who have it successfully removed is 98%, whereas if it's removed after it's already metastasized, the 5-year survival rate is less than 20%. And to identify a melanoma, they use the ABC guidelines. So A for asymmetry, it just has an irregular shape or a non-uniform shape. B for borders, which are indistinct. C for color, and this means that melanoma isn't one color throughout. And D for diameter. Melanoma tumors have a much larger diameter, or just a larger diameter than normal moles. So here are some pictures of the three types of skin cancers. A is basal cell carcinoma, B is squamous cell carcinoma, and C is melanoma. And remember, they use the ABC guidelines for melanoma, but not necessarily these other two. And here, the words above the picture of melanoma tell you exactly what ABC stands for. Okay, now we're at our last type of carcinoma, and that is prostate cancer, which occurs in men, most commonly after age 50. Prostate cancer is quite common. It typically affects men over age 50, so men of this age need to have either a digital rectal exam or a prostate-specific antigen PSA blood test, or both, annually. And the symptoms of prostate cancer include difficulty or inability to urinate, blood in the urine, or pain in the pelvic area. Symptoms for an enlarged prostate are the same as for a bladder infection, so it's good to get those things checked out if you're having them. However, prostate cancer is a slow-growing type of cancer, and it's so slow, in fact, that many men diagnosed after age 65 or 70 may choose not to undergo any treatment just because they're probably going to die of something else before prostate cancer ever gets them. And the treatment for prostate cancer is simply removal of the prostate gland, which is typically pretty successful, radiation, and hormone therapy. The 15-year survival rate is 75% in the U.S., so I would say these treatments are pretty effective. Procedures have also been improved so that nerve functioning, urine control, and sexual abilities are preserved. So now we're going to move on from all of those types of carcinoma and talk about leukemia, which is a cancer of the white blood cells. Leukemia can strike anyone at any age, and even children are susceptible, and there's several different types of leukemia. Typically, a diagnosis of leukemia involves a diagnosis of exclusion, which means that it's diagnosed after tests have been performed and we've excluded all other possible conditions. A definitive diagnosis can be made after a blood test and or a bone marrow biopsy. Leukemia has some pretty non-specific symptoms, which is typically a bad thing when we're talking about cancer, and these include fatigue, weight loss, frequent infections due to a suppressed immune system, basically because the white blood cells are no longer doing their jobs and have been outcompeted by the leukemia cancer cells. Another symptom is bone pain, which I don't really understand why your book forgot to mention that. We're not sure exactly what causes leukemia. Ionizing radiation and or benzene exposure, benzene is a type of carcinogen, have been implicated in the past. And another cause of leukemia may possibly be a virus, as is the case in acute lymphoblastic leukemia, though no specific virus has actually been implicated yet. The treatment for leukemia is chemotherapy to destroy the cancerous cells, often followed by a bone marrow transplant to replace the dead cells with healthy cells. And this is because it's kind of difficult not to kill tons and tons of healthy cells when you're killing the cancer cells. So they need to replace this so the patient doesn't suffer any more problems related to not having enough leukocytes. So here are some slides which are comparing leukemic cells to the normal leukocytes found in the body. And the most obvious difference, just looking at these, is that these leukemic cells have giant nuclei. And typically, leukemias are named after the types of cells they affect, such as, for instance, in lymphoblastic leukemia, we're talking about lymphocytes as opposed to some other type of leukocyte. Lymphoma is a cancer of the lymphatic tissue. This form of cancer attacks the lymph nodes specifically. There are two main categories of lymphoma: Hodgkin's disease and non-Hodgkin's lymphoma. And both of these result in enlarged lymph nodes, most frequently those in the groin, armpits, and neck. The other symptoms include intermittent fever, weight loss, and night sweats. And the same with leukemia. Diagnosis is made after excluding more common causes of these symptoms. A weakened immune system from HIV or human T-cell leukemia lymphoma virus, or from immunosuppressive drugs which decrease the immune system's ability. All of these increase the risk of developing lymphoma. And treatment for lymphoma consists of high-dose radiation treatment or chemotherapy, sometimes followed by bone marrow transplant, for essentially the same reason they do it in those who suffered from leukemia. And they may use antibiotics in the future, or rather the use of antibiotics might be a future treatment for lymphoma. Now we're going to talk about one of the scarier types of cancer, brain cancer. And these are typically infrequent, but are usually fatal after a short time. And brain cancer is typically not a primary cancer, which means that the cancer cells didn't start out as brain cells. And brain cancer is typically a sign of metastatic disease, which has spread from a primary cancer of the breast, colon, or lung. So here in these primary cancers, all of these started out as either cells in the breast, colon, or lung. Brain cancer is difficult to treat, as the tumor must be completely removed from the brain to maintain the same level of brain functioning. So apparently removal of a brain tumor can disrupt normal brain functioning, which is never good. This picture just shows the types and frequencies of certain cancers seen in the U.S. in both men and women. So you can just look at this as a sort of a reference diagram for different types of cancers in the different sexes. The incidence of most cancers increases with age, and this is because as life expectancies increase, there has been an increase in the number of new cancer diagnoses and cancer deaths each year. The long-term survival rates for many cancers continue to rise, and the seeming lack of progress in defeating cancer is actually due to an increasing proportion of cancer causes being diagnosed in the elderly. And here's a quote from me personally: "If you live long enough, chances are you're going to get cancer at some point." Also, medical science has made incredible advances in treating this series of diseases, cancer, and there are many exciting new techniques on the horizon, and we're going to talk about some of those in the next section. Section 11.3, cancer can be diagnosed and treated effectively. Diagnosing cancer requires many tools, and here are 4 types of methods used to diagnose cancer. These are screening tests, imaging techniques, tumor enzyme tests, and genetic tests. There are several routine screening tests. These routine screening tests include manual self-tests, such as a breast self-examination for women and a testicular self-examination for men. A lot of times people will find irregular lumps in these organs. Most of these will be benign, but every once in a while they might be cancerous. And these tests are easily performed while taking a shower or looking in the mirror. And another type of test you can do on yourself is a visual self-examination, where you check yourself for skin cancers or suspicious moles. And here we're just going to check the neck, face, scalp, behind the ears, forearms, and hands for peculiar looking moles. A physician or other healthcare provider can also perform manual tests, and these are typically performed during routine physicals. These will include a manual examination of the lymph nodes of the neck, armpit, and groin. For men, this would include a testicular exam, digital rectal examination of the prostate for men over 50, as much fun as that sounds. And for women, this would include a manual breast examination, manual pelvic exam, which would need to be performed every 3 years for women between 20 and 40, and annually for women over age 40. And for both men and women, they would also perform a digital rectal exam and card smear for occult blood in the stool, which would need to be performed annually after age 50. So here they're just looking for colon cancer for the most part. And there are some types of screening tests that require the use of instruments. These include pap smears for cervical cancers, which should be performed annually beginning at age 18 or at the onset of sexual activity. And then two other types of instrument tests are the colonoscopy and the sigmoidoscopy, which is essentially just like a colonoscopy. They just examine the first portion of the colon, and they do this to screen for colorectal cancer. This should be performed every 5 to possibly 10 years, or maybe even more often if there is a history of colorectal cancer in your family. Here are just some illustrations of how to perform a self-examination for either breast cancer or testicular cancer. They're pretty self-explanatory. I don't know exactly why your book wanted to talk about mammograms twice, but here they are again. And they are a key diagnostic tool for breast cancer. And like I said, a mammogram is a special X-ray for breast imaging. Mammograms can detect cancerous tumors which are too small or too deep in the breast tissue to be detected in a manual examination. It is recommended that women have their first mammogram between ages 35 and 40, with annual mammograms beginning at age 40. And it's important to have a mammogram because breast cancers diagnosed at an early stage can often be removed in a procedure that spares essentially all the breast tissue. There is a wide variety of imaging techniques used to diagnose cancer, and these include X-rays, which can show large anomalies and masses in soft tissue, such as those associated with lung cancer. There are computerized axial tomography, CAT, or CT scanning, and in this technique they use computerized analysis of continuously scanning X-rays to create a cross-section of the area being scanned. And this creates a three-dimensional image which a physician can look at and infer information about a tumor's size and position relative to the body organs. Another type of imaging technique is magnetic resonance imaging, or MRI, which is helpful in pinpointing and identifying tumors in the connective tissues as well as tumors of the brain or spinal cord. And then there's ultrasound. In this technique, they use high-frequency sound waves which bounce off tissues of different densities at different rates, and this helps to distinguish between healthy tissue and tumors. Ultrasounds can provide a visualization of the size, shape, and location of tumors in the prostate, ovaries, kidneys, pancreas, and intestinal tract. Here's an X-ray of someone's liver, which shows that they have liver cancer, and it says that the red here is the liver, whereas the lighter patches of red with the purple shading, it looks like, in them, is the actual liver cancer. Tumor markers and genetic tests can be used to diagnose cancer. So tumor markers are chemicals produced by the body in response to the development of a tumor. Their presence indicates the presence of a tumor. And an example of one of these tumor markers used to diagnose cancer is prostate-specific antigen, or PSA. And this is produced by the prostate cells. And this is actually the only tumor marker that is used for diagnosis, though they might start using others for diagnosis in the future. Other tumor markers can be used to determine whether certain cancers have spread or reoccurred after the initial treatments. Genetic tests can only be used to determine if an individual has a predisposition for a specific kind of cancer. They cannot determine the presence of a growing tumor. And two examples of mutated genes which would indicate a predisposition to breast cancer are BRCA1 and BRCA2. Released substances can be used for DNA analysis. So what we're talking about here are things like urine and feces. Indicators of bladder cancer can be found in cells floating in the urine, and indicators of colon cancer can be found in excreted cells in the feces, and also indicators of lung cancer can be found in cast-off cells released in sputum. Or I'm not sure exactly what they're saying here, but I'm guessing it's whatever they cough up. Now we're going to move on from diagnosis and talk about cancer treatment, and this is a multi-stage process. Cancer treatment has long focused on either killing or removing the primary tumor and then attacking any metastatic tumors. There are 3 standard treatments for cancer. These include surgery, radiation therapy, and chemotherapy. And these are called brute force or sledgehammer methods just because they're pretty brutal compared to some of the other newer methods of cancer treatments. In addition to these, there are also at least 3 newer, more delicate forms of cancer therapy. And these can cripple the proteins that promote the cancer. They can give you drugs to cut off the blood supply to the tumor. These are called anti-angiogenesis drugs. And then there's immunotherapy, where we attack the tumor cells with specifically designed or selected immune cells or antibodies. And at the moment, this includes experimental treatments like cancer vaccines and gene therapy. And these 3 types of methods are called intelligent methods, as they are capable of targeting only cancerous cells or blocking cancer cell growth signals, unlike the sledgehammer methods. Surgery has been the first cancer treatment available to us, and it is still a key tool in fighting cancer. Surgical removal of cancerous tumors was performed even before the discovery of anesthetics. And here in surgery, we simply remove the tumor from the body. In order for surgery to work best, the cancer must be detected early and be in situ or confined to one small space. It hasn't metastasized. Surgery can be a very successful method for getting rid of early detected basal cell and squamous cell skin cancer, as well as certain colon cancers. However, most cancer specialists or oncologists will usually recommend follow-up radiation therapy or chemotherapy to ensure that they kill any cancer cells that may not have been successfully removed by surgery. Radiation can be deadly to dividing cells. Radiation was first employed as a medical aid soon after the discovery of X-rays in 1895. And radiation damages DNA, which typically prevents cellular divisions and leads to the apoptosis of the affected cells. And radiation therapy is generally used if the cancer has spread from its original site but is still localized. And this is a type of secondary treatment after surgery. Radiation therapy is used as the primary therapy for those cancers for which surgery is especially difficult or undesirable. So it'd probably be something like leukemia or lymphoma, where they can't just surgically remove all the cells. However, radiation therapy does not distinguish between cancer cells and the cells of healthy tissue. It just kills all of them. And it may or have various localized side effects, such as hair loss, irritated skin, and even blistering burns at the treatment site. May also cause fatigue and nausea, and could potentially lead to more cancer due to the damaging of your DNA. Chemotherapy disrupts cells throughout the body, and this is the treatment we typically associate with cancer. In chemotherapy, they use compounds that specialize in killing fast-growing cells, so this is a good method for treating cancers which have spread. Chemotherapy interrupts cell growth throughout the entire body in both cancerous and healthy cells. So it actually kills both cancerous and healthy cells. However, the hope is that the growth of the cancerous tumor will be stopped before the drug causes death of the healthy organs. Chemotherapy is either given orally or by infusion. The drugs used in chemotherapy travel throughout the body and damage rapidly dividing cells wherever they can find them. However, some of these damage cellular DNA. Others interfere with DNA synthesis, and some attack cancerous cellular processes. So that's the damaging the DNA is kind of a double-edged sword, because you kill the cancer cells, but you could potentially cause cancer in the other body cells due to damaging the DNA. There are different drug cocktails which are created to attack different types of cancers. Usually they use many different drugs in chemotherapy, and this also prevents the cancer from developing a resistance to one particular medication. However, there are several side effects of chemotherapy, which include nausea, lack of appetite, fatigue, hair loss, dry mouth, anemia, and reduced immune system functioning. The last two are pretty bad symptoms, and someone with cancer might actually experience other side effects depending on what drugs they're being given. Bone marrow transplants are another tool in treating cancer, and these are typically performed in individuals with either leukemia or lymphoma, and they can be performed in conjunction with chemotherapy or high-dose radiation therapy, as both of these therapies destroy the fast-dividing bone marrow cells. We're essentially just making sure the patient has enough bone marrow in their body. And ideally, bone marrow transplants are undertaken using a process called autotransplantation. So, in autotransplantation, bone marrow is located within the skeleton of the cancer patient, and this bone marrow is removed before cancer treatment. The stem cells from the bone marrow, which will form red and white blood cells, are harvested and stored. And then after radiation therapy or chemotherapy has destroyed the patient's remaining bone marrow cells, the stored stem cells are transmitted back into the patient so that they can grow new cells. So one big advantage of this is we're not really concerned with rejection of these cells because they came from the patient's own body. And like I said, this procedure is generally performed for patients with cancers of either the blood or bones. Now we're going to talk about the intelligent methods of cancer treatment, and we're going to start with immunotherapy, which boosts the immune system. The goal of immunotherapy is to boost the immune system in an effort to help it fight the cancer more effectively. And this is done in one of two ways. We can increase the amount and activity of certain types of natural killer cells, and we can assist in the killing of the cancer cell through creating vaccines against the cancer. So essentially in this method, we're wanting to use the cells already within the patient's body to fight the cancer. Adding such compounds as interleukin-2, interferons, and tumor necrosis factor, which assists in killing tumor cells. All of these will assist the body's natural immune cells in fighting cancer. Anti-angiogenesis drugs can starve a tumor. So when cancerous tumors reach a certain size, about 1 to 2 million cells, this is when angiogenesis begins, and the tumor cells do this because the tumor must have its own blood supply to grow past this size of 1 to 2 million cells, and it accomplishes this by forming new blood vessels which feed blood into the tumor. And in theory, once the tumor runs out of nutrients, it should shrink and die. So without a blood supply, the tumor cells cannot remove their waste products, resulting in toxic buildup and further cell death. So with these anti-angiogenesis drugs, we're essentially just removing the tumor's ability to perform angiogenesis so that we starve the cells to death. And researchers are currently studying a number of drugs which are designed to stop the process of angiogenesis. Another type of therapy that holds great promise is genetic therapy. And gene therapy is the introduction of genetic material into an organism in order to prevent or correct a disease or disorder. And this is typically ideal for diseases that are caused by only a few genetic mutations, and they're not really ideal for the treatment of most cancers. A large number of genetic mutations would need to be corrected in most cancers to get rid of the cancer, and there's a pretty complex relationship between the mutation and later promoters of cancer. However, despite these problems, scientists continue to research gene therapy as a treatment for cancer. These new techniques are wanting to use viral delivery of DNA to tumor cells, and these show some promise, though I really wouldn't consider gene therapy to be one of the big treatments for cancer in the future. The two final types of cancer treatment that we're going to talk about are magnetism and phototherapy, which are still in the early stages of experimentation. So in magnetism, powerful magnets are being experimented with in an attempt to target the chemotherapy drugs more precisely. And in experiments with liver cells, tiny metallic beads coated with chemotherapy drugs are injected into the patient, and then these powerful magnets are positioned over the tumor with the intent that their magnetic force will pull the drug-coated beads deeply into the tumor tissue. And then phototherapy involves lasers and light-sensitive drugs, which are being used to insert chemotherapy deep into tumors embedded in organs. So the same sort of thing here, we're just using lasers and light-sensitive drugs. So first, light-sensitive drugs are injected into the bloodstream and eventually travel to the tumor. And initially, these light-sensitive drugs are inactive. However, a laser light is directed at the tumor and set to a particular frequency that sets off a chemical reaction in the drugs, which enhances their ability to kill tumor cells. And it's pretty easy to see that these are still experimental, because honestly, some of this sounds like it came straight out of Star Trek or something. Personal choices help fight cancer. There is a lot of evidence that our personal choices increase our risks of getting cancer. As scientists learn more about cancer, people who develop cancer are able to live longer, healthier, and more rewarding lives after diagnosis and treatment. And it is important that we all support research into cancer prevention and treatment, as it may save our lives. So that's it for this module's lecture slides. In the next module, we're going to talk about the reproductive system in both women and men, and then we're going to go on and talk about pregnancy also. And that will be my longest set of slides, I think totaling 85 slides. So just be ready for that.